Bismillahirrahmanirrahim. Uh, Myself, Dr. Waqas Ashraf. I'm working as assistant professor at the Department of Microbiology, and today I'm going to present a topic which is related to my NLP project titled Robust Molecular Detection of Silent Circulation of FMD and PPR viruses in Cholistan as a model for disease-free zone. And relevant to this project, my presentation is pros and cons on reverse genetics for the silent circulation of foot-and-mouth disease virus in Cholistan. The main important aspect which I want to propose is that through this presentation is how we can address the silent circulation of the FMD virus, because main reason that even after vaccination the animal can contract virus, and especially even in cases when animal is recovered from the foot and mouth disease infection, still the virus is able to survive in the pharyngeal fluids, in the pharynx. And ultimately, there are chances that the virus can be passed on to the susceptible ones, and ultimately the virus will remain circulating in the population. So address this problem, I have proposed this idea. So first of all, what is genetics? So genetics is the study of genes, genetic variation, and heredity in organisms. So genetics helps to explain how traits are passed from the parents to their young ones. So in conventional genetics, we have different concepts relevant to genetic dominance, Mendel's law of segregation, then Mendel's law of independent assortment. There is polygenic inheritance, and then there are certain sex-linked traits, and of course, all the genetics is related to genes and chromosomes, and the expression of genes in the form of protein synthesis, and then how these traits they are transferred to the newly dividing cells, and how these traits they are transferred from the parents to the offsprings. However, in case of virology, the we are concerned about the viral genome when we consider the concept of genetics in viruses. So there are a lot of mutation rate in case of RNA viruses as compared to DNA viruses, because the viral genome they are of diverse type, and viruses they keep on changing throughout time, and of course by looking at the rate of mutation we can find out how long the time has passed and what kind of changes the virus has got through mutations. So in the case of RNA viruses, the mutation rate is 1 per thousand to 1 per 10,000 nucleotides, and that's why the RNA virus they have got greater potential to mutate and evolve over time in comparison to DNA viruses. So in case of viruses, we can have recombination. There can be independent assortment. There can be incompletely linked genes. Of course, there will be phenotypic variation from recombinations. And again, yes, the vaccines can be prepared by utilizing this phenomenon of recombination. So the as we know, the Baltimore classification pattern, that is the central point of that classification is the messenger RNA. And of course, the virus they have to prepare the proteins once they have to once they try to replicate in the host cells, they have to pass through the process of mRNA synthesis and subsequently followed by a protein, and then assembly of the virus once the new virion they are synthesized. So based on the Baltimore classification, we have got certain classes, including the viruses which have plus sense RNA, then we have minus sense RNA, genomes within the virus, and then we also have combination of both plus and minus sense RNAs within the same virus, like reovirus, rotavirus. And for minus sense, we have got influenza and PPR virus, the virus which I am working on. One is the PPR and the second one is the FMD. One is negative sense and the other one is positive sense. So by looking at this diagram, we can easily assess how many diverse type of viral genome they exist in different viruses, and accordingly we can assess how complex they can be, and accordingly how difficult it can be to control the viral diseases. Once a virus exists in our environment, it is not going anywhere. It will be staying. So somehow we have to, first of all, we always try to prevent the occurrence of vaccine through vaccination, and then we can also go for certain targets to develop certain antiviral drugs. But of course, then the antivirals, they are very costly and difficult to afford. So mostly we go for vaccines. So for the FMD virus, it is a plus sense single-stranded RNA virus, and it has to synthesize its minus sense intermediate copy of RNA to prepare the plus sense genome copies for its new virions, and accordingly, however, the protein, they can be synthesized from the plus sense genome. So once the proteins can be synthesized from the plus sense genome from the parent origin, and subsequently the new plus sense genome, they can also be utilized to prepare the protein copies, and ultimately both will assemble and prepare the new copies of the virus. So this is the tertiary structure of RNA. RNA is not a single, even if it is single-stranded, it is not going to exist in a straight single thread, but it will be having some scanty structure, some overlapping regions, which may be somehow complementary to each other. So preparing stem loop structures, and ultimately such kind of structure, they also play the role in the regulation of certain gene expression and the efficiency of the viral genome to be copied in the host cells. Regarding FMD virus, its genome, it's belong to the Bicordinaviridae family, and the genus ectovirus, its genome is having 8,004 nucleotides, having open-ending frames for the structural proteins as well as non-structural proteins. And regarding the difference between the structural and non-structural proteins, for the vaccines, we generally go for structural proteins. And the reason is that the antibodies which are being synthesized against this protein, they are supposed to function as neutralizing antibodies. So once we target to prevent the entry of virus into the host cell, we are definitely going for the structural proteins. Or the non-structural protein, they have also a certain role. And however, when we are interested to develop certain vaccines which can be linked certain diagnostic viruses, especially in those cases when we want to differentiate between vaccinated and naturally infected animals. So this approach is quite useful, and this fortunately exists in case of FMD as well. That we have got purified vaccine available in which only structural proteins, they induce the immunity against the virus. However, the non-structural proteins, they can be detected through ELISA to differentiate whether the animal is infected with some kind of wild type virus, or the antibodies, they are due to the vaccination actually. So this is really a good strategy when we want to evaluate our vaccination campaigns, how much we have been successful in reducing the virus burden in the population, virus circulation within the population. Sorry. So this is the arrangement of FMD viral genome. Starting from the VP4, VP2, VP3 from the left side to right side, we have got first structural proteins and then non-structural proteins, and then again to the function of the RS, how it is going to control the host cell machinery to synthesize only viral genome and not the host own proteins. So once the FMD virus it infects the host cell, it will capitalize the host machinery, and ultimately cell will not be able to synthesize its own proteins properly as it works during the normal physiological process. But once the virus has infected the cell, the host protein synthesis own mechanism will be compromised, and it will be starting synthesizing only the viral proteins. And again, we have got certain protein which we use for the serotyping of the virus. So the FMD virus outer shell, which is capsid, made of 60 repeated units, and again for VP1 that forms a pentamer, VP2 has trimer, and VP3 has trimer. So this is the structural description of the surface proteins, how they are arranged on the wall capsid. So FMD is a small ecocidal virus which is 30 nanometer in diameter, comprising of 60 copies of each of the four structural proteins, starting from VP1 to VP4. So here the VP1 is in blue, VP2 is in green, and VP3 is in red. So here we can easily see how these proteins they are arranged. However, VP4 is not such visible in this diagram. That is actually embedded on the inside, and that is actually interacting with the RNA. And this has been proposed that a certain effort has been made to synthesize only the viral capsids without the viral RNA, like VLPs. However, the actual significance, the actual potency to induce the immunologic response that is relatively not up to the level which is required, as in case of whole virus. And the reason has been proposed that RNA is going to interact with the stability of this whole capsid, and ultimately the development of neutralizing antibody that requires the interaction of the surface protein with the viral RNA. In addition to that, we can also target certain protein like HSP90, which is of course required for the by the FMD virus to stabilize its protein structure and ultimately assemble, and it has also been proposed as a target for certain drugs. And then within the this surface protein, we have got a VP1GH loop, which has been proposed to be critical when we have to design certain vaccine based on these structural proteins. So the next is if we want to prepare a vaccine, so then first of all we should know which kind of antigenic or immunogenic epitopes they are existing within these proteins, especially when we are going to apply the technique of reverse genetics. So this was a really good article mentioning certain immunogenic epitopes within different various serotypes. Like here we have got VP1 gene, VP2 gene, and within various serotypes, which kind of antigenic epitope they do exist within these protein structures. And again, this diagram is indicating the conserved regions within these structural proteins among the various serotypes. So once we have we analyze the sequence of viral proteins, then we also go for the analysis of the viral genome. So this will be an approach which is in the forward direction of genetics. Once we have determined the sequence, and then the certain we have predicted the antigenic epitopes and how the protein they are interacting with each other, and how the virus is interacting with the host cells. By utilizing that information, we can also go for reverse genetics. That means we are utilizing the genetic information to reorganize it in such a way that its phenotypic traits, which are of our own interest, they can be developed. So this is the basic concept of how we go from the phenotype to the genotype from forward genetics, and then how we utilize the genetic information to make some changes or rearrange them to develop certain traits which are of our own interest, especially in case of vaccines. So conventionally, the reverse genetics approach it includes gene silencing. We can go for target gene disruption by homologous recombination, and then certain insertion and mutagenesis or transplant-mediated mutagenesis can also be done. However, in case of virology, we can use some other approaches for reverse genetics, including RNA polymerase 1. Then we can go for plasmid-based reverse genetics. Then bidirectional polymerase 1 and polymerase 2 transcription system can also be used. Then they can generate RNA polymerase 1 constructs, and we can also use cell lines for transfection as well. So FMD stores genetic material in ribonucleic acid as opposed to the deoxyribonucleic acid, and then as opposed to the DNA, and making it difficult for scientists. Of course, for certain viruses which are having RNA, we first need to convert it into DNA to apply this reverse genetic system, because directly RNA manipulation is relatively difficult. However, new approaches or RNA synthesis they are in the field now. So by using this technique of reverse genetics, we can go for this is a full genome copy of the virus, and we have to convert the RNA into cDNA by using in vitro transcription to get the infectious virus clone. Here are certain examples in which the sense of, for example, sense of influenza virus and impetus from an old enzyme by using the RNA polymerase 1. In case of influenza and in case of COVID, there has been certain set of protocols which have been developed, which have been proposed and even being applied as well to develop attenuated vaccines of the COVID virus. So one approach is using the RNA polymerase 1 gene. So we have got the three types of RNAs, RNA polymerase within the cells. So RNA polymerase 1 synthesizes a pre-ribosomal RNA, which measures and will form the major RNA sections of the ribosome. Then we have got RNA polymerase 2 that synthesizes precursors of mRNA and moves microRNAs and small RNAs. Then RNA polymerase 3 that is going to synthesize transfer RNA and ribosomal RNA and other small RNAs found in the nucleus and cytosol. And similarly, 4 and 5 they are found in plants and they make the interfering RNA within the plants. However, in case of when we are going to construct, for example, our gene, so we can use this set uh, where the ribosomal RNA gene cluster exists. We can replace it with our own gene of interest, for example, viral RNA to synthesize its copies. So once the plasmid has been synthesized and cloned, it can be transported into cells to prepare the intact viral particles. So for certain viral proteins, we can use polymerase 1 and polymerase 2. For example, if we make a construct which is having a double-stranded DNA on the viral RNA copy, on one side we have got polymerase 2 gene transcription machinery, on the other side, on the opposite strand, we have got polymerase 1 gene transcribing machinery. So ultimately what we will get, we will be having both the plus and minus sense viral RNA copies, and from those new viral RNA can be synthesized the new copies of the viral RNA. For example, if that is plus sense, that can be synthesized, and in the same it can be the case with the negative sense RNAs. And in addition to that, the plus sense RNA will act as a messenger RNA to convert the information, genetic information, to certain protein form. And once this plasmid has been synthesized, it can be transported into the cells, and when it will be translated and copied, so we will get the infectious virus particles. And in case of foot and mouth disease especially, we can utilize this strategy once we know the certain epitopes which are immunogenic in the structural protein. They can be cloned, and ultimately we can make a viral particle which is having these structural proteins in the attenuated viral particle, which will not be causing any disease. However, it will be inducing immunity. So definitely it will be a kind of a virus which is not supposed to cause any disease, and the main target can be the induction of the IgG based immunity as well as the mucosal immunity, which is mainly due to the IgA. And the proposed idea is actually to produce such a vaccine which can stop the existence of virus in the pharyngeal fluids, because that can be a source of the persistence of virus in a population, and ultimately slow leakage to the non-target host, which, for example, sheep and goat, and ultimately they can also pass on the virus to the healthy, to a susceptible population which may not be previously exposed. So in that case, by utilizing this strategy, we can stop the chain of virus persistence in a population. Currently, the vaccine they are which are being used, there and that is the killed vaccine, and ultimately the animals they are somehow prevented. They don't contract. They are saved from the disease. However, they can contact the disease virus, wild type virus, and it may persist in their pharyngeal fluids, and ultimately they can pass it on to the susceptible ones. However, if we have got such a vaccine design which can induce both humoral as well as mucosal immunity, so that can be a better approach to address the silent circulation of virus, in which the virus that that is circulating without showing any particular sign, but it is residing in the pharyngeal fluid and ultimately in the non-target source, for example, sheep and goat. that may not be manifesting a very clear signs of FMD. For example, only lameness is sometimes indicated in case of foot and mouth disease. So in that case, yes, the persistence of virus can be prevented if we design such a vaccine. For example, I was listening to a seminar, and people from Plum Island, they have designed certain vaccines which are able to, in the challenge study, they were able to prevent the occurrence of on the onset of infection, and there was no virus detected in the pharyngeal fluids as well. So with this, thank you very much. And this was a presentation on certain idea how we can stop the silent circulation of virus in our population by improving our vaccine design by utilizing this strategy. Thank you.